Hi everyone, I'm Elizabeth, and today we will be talking about the DNA structure which was discovered by scientists Watson and Crick in 1953. DNA, also known as deoxyribonucleic acid, is a double helix structure that is found in all forms of life. DNA is composed of nucleotides. They contain three important components, a sugar, base, and phosphate. The phosphate attaches to the sugar at the 5' carbon. This attachment between the phosphate group and the sugar is known as the phosphate backbone, and it is connected by phosphodiester bonds. This connection between the ribose carbons gives DNA its negative charges. The 1' carbon attaches to the base from the sugar, linking the two together. The 3' carbon bonds to the next 5' carbon of the phosphate. And finally, the 2' carbon distinguishes DNA from RNA. So as you can see, each ribose carbon has a specific job to do. Now let's discuss the nitrogenous bases. They are split up into two groups, purines and pyrimidines. Purines consist of adenine and guanine, and pyrimidines consist of cytosine and thymine. These bases do something very cool known as base pairing. This is when adenine binds to thymine and guanine binds to cytosine. These bases bind and stack on top of one another, which is known as base stacking. Here is an image I illustrated so you can visualize what base stacking in DNA would look like. As you can see, these strands of DNA are anti-parallel to each other, meaning that it is read from 5' to 3' and the other is from 3' to 5'. These nucleotides are connected by phosphodiester bonds. They are also connected by hydrogen bonds, but hydrogen bonds are weak in comparison, and it should be noted that they are not the main force holding the strands of DNA together, but they do help the bases bond to each other. Base stacking is the main force stabilizing DNA double helix. In the model for DNA structure, there are two components known as major and minor groups. The major groups form when the phosphate backbones are far away from each other, whereas the minor groups form when the phosphate backbones are close to each other, as shown here. I bet you thought only humans could be left-handed or right-handed. Nope, DNA can be left or right-handed too. While right-handedness is most common, there are instances of left-handed DNA too. Here we have an example of a left-handed DNA and a right-handed DNA. An easy way to tell which is to draw an imaginary line starting from the bottom, going to the top through the DNA. I have drawn a dashed line to demonstrate this, but notice that the right-handed DNA dashes go towards the upper right corner and the left-handed DNA dashes go towards the upper left corner. This is an easy to remember method that helps me tell which DNA is left-handed and which DNA is right-handed. Now I would like to talk about salt concentration and its effects on DNA. Salt ions neutralize negative charges in DNA. They bond to the phosphate backbone, and what this results in is a more stable strand of DNA. This raises the melting temperature of the DNA. The more salt ions present in this strand of DNA, the higher the temperature needed to cause denaturation of the DNA. When denaturation occurs, hydrogen bonds are broken and the double strand helix becomes a single strand.